Our last topic for this unit is viruses, and the reason why we talk about viruses in this unit is because we're talking about living things, and even though viruses are not really considered living, and we'll talk more about that later, they do affect everything that is living. So to start off, just to talk about viruses, you're going to answer 5 questions on the first page, and these are the same pictures you would be looking at. So as I kind of talk about the viruses and you look at the pictures, feel free to just answer the questions along the way. So starting first, viruses are extremely diverse, and they are very specific as to what they infect. So for example, we have this tobacco mosaic virus. Tobacco we know as being a plant, so this is a virus that would infect plants only. Here we have a virus called a bacteriophage, which, if you see that word bacterio, this would be a virus that infects bacteria only. And then lastly, we have the HIV virus, and we know the HIV virus to be one that infects animals, such as humans, which is what we are. We know we're classified under kingdom Animalia. So again, all viruses are very specific, meaning we cannot get infected by the tobacco virus. So humans are safe from that. Plants are safe from HIV. Bacteria, they're safe from HIV as well. So again, extremely diverse, but also specific as well. Some things that viruses do have in common is every virus has some sort of protein outer coat. You'll notice here in the tobacco, we have what's called a protein coat here on the bacteriophage, and we also have a core protein coat here on the HIV. Other things viruses have in common is they have some sort of genetic material inside. In the case of tobacco, the genetic material is something called RNA. It's a lot like DNA. We'll talk about the similarities and differences in another unit. Bacteriophages have DNA, and again, we take a look at our HIV. They also have RNA. So they all have some sort of genetic material. Some viruses will have special parts like the HIV. So we're looking over here now at the HIV virus. They have an outer lipid envelope. Now lipids are fats. We'll talk more about those again in the next unit, but they are just a way for the HIV virus to trick the host cell, in this case it would be human cells, into thinking it's harmless. It will allow the virus inside, and of course once viruses are inside, that's when they can do the damage. We'll talk more about viral reproduction in the next slide. So how exactly do viruses reproduce? This right here is simply just an example. It is not the only way that viruses can reproduce, but the key is in order for reproduction to occur, all viruses need a host. So here we have an example. In your notes, we have this divided into 2 types. We have an A and we have a B. There's 2 different reproductive cycles in viruses. I'm going to start my discussion with cycle A. This is called the lytic cycle. The lytic cycle is also known as the infectious cycle, meaning if the virus goes through this cycle, you will feel sick or the cell will be destroyed. In the example we're looking at here, we're looking at a virus infecting a bacteria. So what would happen is the bacteria would actually die here in the lytic cycle. So starting at step number 1, what will happen is the virus will attach to the host, and you'll notice this blue stuff here, and it will inject genetic information into the host. That is step 1, injecting viral genetic material into the host. Okay, that's it. Step 2. At this point, we can see the genetic material from the virus, the blue stuff, has completely entered the host. So in step 2, the DNA or genetic material from the virus starts taking over the cell. Step number 2. Okay, next we have step number 3. In step number 3 of the lytic cycle, you'll notice, look at all these viruses being made. In step number 3, the genetic material has now caused the cell, the host cell, to make more viruses. So it's making more viruses instead of doing what it's supposed to be doing. And then lastly, in step number 4, the reason why we call this the lytic cycle is because the cell will be now filled with so many viruses, it will lyse or burst open, causing the cell to die, and now more viruses can go and infect other cells. But there is another type of cycle, and that is called letter B, the lysogenic. In the lysogenic cycle, what happens is the virus infects the host, but it stays dormant. Kind of like a volcano is dormant, it is possibly going to explode, but we don't really know when, and that's sort of the case here. So we start off the same. Step 1, inject material. Step 2, here's where a slight change. So the genetic material is now in the host cell, but it doesn't start taking over. We move on here. I'm just going to call this number 5. You'll notice that the genetic material hides itself within the host genetic material, which is the brown stuff. And when it's hiding, it is dormant. And we call that hiding or dormant viral DNA, we call that a prophage. And as long as it's hiding and staying dormant, it is not going to do any damage. We're not going to make any viruses. So now when the cell goes to reproduce and split, which is happening here at step number 6, the host cell is reproducing and splitting. Now every cell from this point on will have this hidden viral DNA inside. And that's it. And that's really what step 7 is showing. Now here, this step 7 looks different from your picture. In your picture, what you see happening is, and I don't have the blue up right now. In fact, here, let me just make this blue. That viral DNA that was staying hidden is now going to come out from hiding. And that's what's going to happen in step number 7. Once that viral DNA comes out from hiding, it can then loop back into the lytic cycle. So, application. You may have all heard of herpes before. Herpes comes in different forms, simplex 1 and simplex 2. Herpes simplex 1 infects pretty much from the waist up. And so herpes simplex 1 is when we are talking about cold sores. So a couple things can happen. You can come in contact with the herpes virus and it can go into the lytic cycle right here. And if it does, you'll get a cold sore because it's infectious. And yes, it can spread and you can give it to other people. Or the herpes virus can enter one of your cells and go dormant. And then when your lip cells are dividing, which they normally do, you have this hidden dormant viral DNA inside. And then all it takes, we're going to look here at step 7, is for that hidden DNA to come out of hiding, that prophage to come out of hiding, and then it can loop into the lytic cycle. So a couple things that happen with lytic and lysogenic. Once in the lytic cycle, it's in the lytic cycle. So it doesn't go from being infectious to dormant. That never happens. But you can have something go dormant and become infectious. So you can go from lysogenic into the lytic cycle. So at this point, you should be able to fill in the information on this page up through letter D. And then you should be able to turn the page and fill in the chart using your old cell notes and any information you learned about this video.